Field Practices, Sampling Design and Census of Marine Communities. Sampling Methods. Quadrats. A quadrat is a square or rectangular sampling unit in which organisms are collected, counted or measured. Quadrats allow a quantitative estimation of biodiversity. Quadrats could be carried out using frames made of PVC, metal, or wood. However, PVC frames are preferable for underwater samplings since they are not damaged by salty water. Size of frames depends on sample size. In general, in the Mediterranean waters, a half-a-meter quadrat is used to study sessile benthos. It is also possible to use quadrats of 25 centimeters for small samples. In case of turbid waters, and very small samples, it would be possible to further reduce the sampling area. Quadrats could be used both for destructive and non-destructive sampling methods. In this slide, a quadrat is used to collect benthic sessile organisms on a rocky shore. Quadrats are used to delimit a surface, when vagal fauna is collected through an airlift system. Quadrats are mostly used for non-destructive samplings such as visual census and photo surveys. During a visual census, the organisms will be detected and identified directly in situ. Scuba divers conducting this kind of survey should be expert in species identification. Remember to bring with you a dive computer to check sampling depth and a dive slate to record identified taxa. Do not forget your frame. Spare pencils also are suggested. This underwater technique is time-consuming. In order to establish how many minutes you could dedicate to collect data, plan your dive activity before going to dive. If you have to collect data at different depths, start from the maximal depth. Dedicate the same time interval to collect data at each depth to standardize the sampling method. If you have an underwater camera, you could take pictures of organisms that you cannot identify. After diving, share your doubts with your dive buddy to solve eventual identification issues. Transcribe as soon as possible collected data to a spreadsheet due to pencil discoloration. Data elaboration would be very fast since the species have already been identified underwater. Quadrats can be used to delimit a surface to be photographed. Underwater pictures will represent a permanent record. This underwater technique is fast to carry out. Moreover, the pictures could be collected by not trained scuba divers. When you are performing a photo survey, place the frame parallel to the wall. Avoid placing the frame in correspondence of holes or protrusions. The sampling area should be homogeneous and with the same inclination. Take the pictures placing your underwater camera parallel to the wall to avoid parallax. Photographic samplings can be carried out on both vertical and horizontal substrates. Take note of substrate slope on your dive slate. A plastic or metal spacer can be interposed between the camera and the frame to avoid parallax. It also will ensure keeping the same distance throughout the sampling. After samplings, photos should be analyzed through an image processing software and species should be identified by experts. Then, data could be transcribed to a spreadsheet to be elaborated. Quadrats should be placed at random intervals along a transect line. See Lecture 01 to see details of random samplings and about the use of random digit tables to obtain random replicates. To perform quadrats, it is possible to use simple frames or gridded quadrats. The simple frames are more suitable to determine density of quite large, countable organisms such as gorgonians, branched sponges, Cymodocea tufts. Even vagal species such as echinoderms could be counted using this kind of quadrats. Simple frames are preferable when fragile colonial organisms should be counted, such as bryozoans or red corals. The density is obtained dividing the total number of individuals of a species per the surface delimited by the quadrat. In the illustrated example, density of the yellow Gorgonian Unicella cavallini is 1 individual per square meter. With this method the organisms are counted but not measured. Therefore, you wouldn't know if the counted organisms are small or big. This could be compensated by considering size classes of the same species. For example, in the case of the yellow Gorgonian, we could count separately larger colonies, medium-sized colonies, and juvenile colonies. Gridded quadrats are more suitable to determine abundance of more or less bidimensional organisms. This method should be avoided to sample fragile organisms, because they could be damaged by the grid. The abundance could be expressed as percentage cover, or frequency. Fragile organisms should not be considered. Percent cover can be estimated by dividing the surface that each organism occupies in the sample area per the total examined area. To estimate percent cover, it is necessary to take pictures of the sampled area. The surface of each organisms can be calculated using an image processing software. Next videos will explain how to use an image processing software to gather data from photos of marine benthos. Percent frequency could be calculated in different ways. Method 01. Percent frequency of a species is calculated by dividing the number of subquadrats where the species occurs, per the total number of subquadrats. For example, species 1 is present in 7 of the 25 subquadrats. Therefore, its percent frequency is 28%. Species 2 is present in 8 of the 25 subquadrats. Therefore, its percent frequency is 32%. Species 3 is present in 8 of the 25 subquadrats. Therefore, its percent frequency is 32%. Exercise: calculate percent frequency of species 4. Exercise: calculate percent frequency of species 4. Percent frequency. Method 02. With this methods, truth really occupied subquadrats will be considered. The number of occupied subquadrats will be divided per the total number of subquadrats. Consider species 1. The species is present in 7 subquadrats, not it does not occupy 7 entire subquadrats. If we splitted species 1, and if we try to insert pieces inside subquadrats, we could see that species 1 approximately occupied 1.5 subquadrats. Therefore, the percent frequency of species 1 is 6%. Exercise: calculate percent frequency of the indicated species applying method 02. Method 03. Consider points where lines intercept. Percent frequency is given by the number of overlaps between species and intercept points per the total number of intercept points. Therefore, percent frequency of species 1 will be given by dividing 3 overlaps per 16 total intercepts per 100. Exercise: calculate percent frequency of the indicated species by using method 3. Exercise: compare your results obtained with method 1, method 2 and method 3. Which was the preferable method? And why? To build a simple PVC frame, you could use PVC electrical conduit pipes of 20 mm in diameter, elbow connectors or T-connectors, steel bolts or steel bolt ties, washers and nuts. When you measure and cut the pipes, consider that the area useful to collect samples is the area inside the frame. Also consider the length of the connectors to obtain the size you want. The exceeding extremities of the bolt eyes could be useful to fix temporarily the frame to the wall. In order to get negative buoyancy, Make holes along the pipes to allow water entering the frame. Indeed, it is easier to handle negative tools underwater. If you want to build a gridded frame, pass a thick thread through the holes. If you need permanent plots to carry out your experiment, you could fix the frame permanently to the wall by using longer screws and marine 2-component epoxy putty. Another way to obtain permanent sampling stations is to permanently fix spikes or pickets to the wall, which temporarily support the frame during surveys. This solution is preferable when the sampling site is in shallow waters, and permanent quadrats could be removed by water movement. Quadrats could also be foldable to be mounted at the time of use. However, this would be time-consuming and there are not so many advantages to use foldable quadrats. You could find an example of using permanent quadrats in the open-source paper Takesito et al. 2013. Look at the plate. Pictures on the left were taken before an intense storm event occurring on December 26, 2008, while pictures on the right were taken after the storm. The storm mainly affected long-living organisms, such as gorgonians. This is another example about the use of permanent quadrats. The two pictures show a portion of a rocky substrate before and after a mass mortality event occurred in the North Adriatic Sea, on 2011. The event, due to pollution, caused a severe disease of several benthic species. One of the most damaged species was the sponge Chondrosia reniformis, that you can see in Figure E. The species died and disappeared in 2011 in the sampled area. Figure F shows the same area photographed in June 2013. You can see that, after 2 years from the disease, the sponge did not recover and the substrate was still bare. Note that permanent quadrats give the possibility to monitor benthic communities over time and to quantify temporal changes in biodiversity. Here, you could find a schematic comparison between samplings at permanent and random stations. Also remember advantages and disadvantages of visual census and photo surveys. Thanks for watching.